Endoplasmic reticulum. We will start with this endoplasmic reticulum. Now, when we talk about endoplasmic reticulum, refer to the this is where is the previous work written? It's there. So now the endoplasmic reticulum, you have to remember, if you refer to the diagram that you have drawn, endoplasmic reticulum refers to what? A network of tubular structures. No? The endoplasmic reticulum, if you understand, I talk about the diagram of the animal cell. How did we draw the endoplasmic reticulum? If this is the nuclear region, then how should we draw the endoplasmic reticulum? A network of tubes like this. It is a network of tubes like this, a network of tubes like this, and this network of tubes has got certain type of functions. Already you have drawn all these. This is endoplasmic reticulum. So I am talking about endoplasmic reticulum today. Now, this endoplasmic reticulum, if you remember, we had already drawn this, which are nothing but in the form of some tube-like structures, in the form of some tube-like structures, in the form of some tube-like structures. And these tube-like structures, if you check the diagram you have drawn, that these tube-like structures can be either like this, or these tube-like structures can be this way, where they may generally have some another organelle attached to their outer surface. And what are their functions? So this is what we are going to study now. Endoplasmic reticulum, say suppose we talk about the structure first of all. The structure, the types. So what about the structure? What about the structure? The endoplasmic reticulum is a network of tubes. It is a network of tubules or a network of tubes. So what is the structure of the endoplasmic reticulum? It is a network of tubules. And always to remember, these tubules basically start from the nuclear envelope. Always starts from the nuclear membrane. It will always start from the nuclear membrane, and it will always span over the entire width of the cytoplasm. It will span over the width of the cytoplasm, sometimes ending up in the cell membrane and sometimes not. So sometimes they will end up in the cell membrane. Now I may continue drawing this like this and show that yes, it is completing and uniting with the cell membrane. Sometimes it may be like this, not necessarily every time it should be this way. So it may be like this, it may be like this also. But whatever it is, they will always start from the nuclear membrane. When we talk about the types, the endoplasmic reticulum, do they have types? Yes, we are talking about RER. What is RER? Rough endoplasmic reticulum. So why do we say, why do we name it as RER? Because the membranes are generally having a whole lot of organelles attached, which give a rough appearance actually. So RER actually means endoplasmic reticulum. RER actually means endoplasmic reticulum plus ribosomes. So endoplasmic reticulum with which ribosomes are attached. So if I write endoplasmic reticulum with ribosomes attached. Endoplasmic reticulum with ribosomes attached. So if I'm talking about endoplasmic reticulum with ribosomes attached, then I'm talking about RER. What about the other one, SER? So when I talk about SER, so it is only endoplasmic reticulum, no ribosomes attached, only endoplasmic reticulum, only endoplasmic reticulum it is. So now that we are talking about the types of endoplasmic reticulum, so these are the two types. Endoplasmic reticulum, we are supposed to remember, endoplasmic reticulum has got two types. It can be smooth, it can be rough. And what is the major difference? Understand. Next is when I talk about endoplasmic reticulum. Okay, one more feature. Let us talk about the rough and the endoplasmic reticulum. What about their location? Where are they found? If I have to remember.
Near the nuclear membrane.
No, location of the RER. Location of the RER, I would say, majorly. RER would be majorly present in large number in the muscle cells. Muscle cells have got large. Now, in a cell, not necessarily there should be equal number of RER and SER. In a cell, there may be more number of RER. In another cell, there may be more number of SER. It's like that. So if I have to remember one example, if I have to mention one example of the RER, then we can say that RER is majorly present in large numbers in the muscle cells. What about SER similarly? SER, we would say that it is majorly present, location of the SER majorly present in the liver cells. And there's a reason why. Based on their function, basically, if I have to understand that why SER, we say that it is majorly present in the liver cells, and why do we say that RER is majorly present in the muscle cell, because regarding with the function. Now we learn the function. So what are some of the functions of endoplasmic reticulum? First, let us try to understand the general functions of endoplasmic reticulum. General function of endoplasmic reticulum is it provides structural framework. It provides structural framework. Endoplasmic reticulum provides structural framework.
And both.
Both. So see here, check here that it is nothing but a set of tubes. It is nothing but a set of tubes, and these set of tubes are spanning the entire width of the entire space of the cell. Because this
can be like easily.
Smooth does not mean soft. Here we are talking about smooth, meaning the membranes are smooth, which does not have. Just imagine, I'm talking about a membrane. Now I'm talking about a membrane which does not have any granular substance. And again, I'm talking about a membrane which has a granular substance. Obviously, this will have a rough texture. This will have a smooth texture. So, based on that, the name smooth and rough. But are we to understand that this is very soft? No, it may not be soft. It is a network of some tubular structures, and these tubular structures are almost spreading up in the cytoplasm. They are almost spread out in the cytoplasm. Since they are spread out in the cytoplasm, they provide a shape to the cell also. If possible, remove this endoplasmic reticulum out, or just cut off the endoplasmic reticulum and just take it out. Then the cell will always, the shape of the cell will collapse. The membranes of the cell will just start collapsing, because it is just like our body, the bones and the cartilage, they give us a proper structural framework. No? Same like that. So that's why we say the endoplasmic reticulum gives a structural framework. Next is what? Endoplasmic reticulum helps in the process of transport. Now, what type of transport, if I have to understand? Endoplasmic reticulum gives us transport. Endoplasmic reticulum brings about transport. Now, to understand transport, let us first of all understand the role of SER and RER. Now, if I talk about RER specifically, RER generally helps in synthesis of proteins. This helps in synthesis of proteins. And what about SER? It helps in synthesis of lipids. It helps in synthesis of lipids. So whenever I'm talking about the synthesis of proteins and synthesis of lipids, synthesis of proteins and synthesis of lipids, meaning RER engaged in the process of formation of proteins, SER engaged in the process of formation of the lipids. Now, say suppose I talk about that this endoplasmic reticulum. Did I draw ribosomes around? I have not drawn ribosomes around. So it is understood that this is SER. And if it is SER, then what it must be producing in the tubules? It must be producing a particular enzyme. And this enzyme may be perhaps, I draw in blue so that you don't get confused. Now this enzyme that I'm talking about, perhaps this enzyme that I'm understanding is something like the lipid molecules that I have written there. Lipids means fat molecules, okay, fat molecules. So it is produced here, and the fat molecules are produced here. Now what will happen to this fat molecule? It is getting produced here. And within short period of time, some small bubble-like structures will develop like this. Some small bubble-like structures from either ends it will develop. And these proteins, these lipids, will start getting contained, will start getting filled up here. Now this lipid molecules will start gathering in these small bubble-like structures. After some time what? After some time these bubble-like structures are getting pinched off, they get separated. these bubble-like structures will get pinched off. They get separated. And now, what are these vesicles or what are these bubbles containing? What are these vesicles called? Is basically these vesicles. These are called vesicles. What are these vesicles? Something like small bubbles. Now pinching off from the membranes. And what are they containing? The secretions, either lipids or proteins. Now I wrote there that yes, this is engaged in the process of formation of lipids. Similarly, this is engaged, if I have to show from here, I should show that this is constantly engaged in the process of formation of the proteins. This is engaged in the process of formation of the protein. Same way. Now this vesicles or the vacuoles which are containing the proteins and the lipids, what will happen to these vesicles? They will keep on circulating in the cell so that the proteins and the lipids that they have prepared, the cell can utilize. This is one function. Also, this can go and move out of the cell. Say, suppose they can go and fuse, they will fuse like this and release the cell secretions outside and release the cell secretions outside, and so that the other cells can receive these molecules. Either the molecules of proteins and lipids will be used up in the cell itself. Now, when will the cell utilize the lipids and the proteins? When a cell is dividing, when the nuclear membrane is getting formed, when the DNA is getting formed. So for the process of formation of all these molecular structures or the membranes, enough of proteins, enough of lipids are necessary. Maybe there's a small damage in the cell membrane. The formation of the cell membrane or the repairing of the cell membrane itself requires enough amount of the proteins and the lipids. So in the cell itself, they have got a lot of functions to perform, or sometimes they will be going out performing their function in certain other cells. So that is what we are talking about as the transport functions, transport of the proteins and the lipids, transport of the proteins and the lipids. Something more that we talk about would be the detoxification. Something more that we talk about would be the detoxification functions. What do you understand by the detoxification functions? Okay, detoxification function. So when I talk about the detoxification, what is the meaning of detoxification? Detoxification basically means breaking down the complex toxic substances, poisonous substances into simpler forms so that the body can release them out. So when I'm talking the detoxification functions, what is happening here? This liver cells, now I talk about liver cells here, no? The liver cells are rich in RER or SER, liver cells are rich in SER. By the way of circulation, so many different types, by the way of circulation of the blood, so many different types of toxic substances must be entering the liver cells every day. Now the question is that how come our body will get all those poisonous substances? Yes, we of course eat food which are rich in preservatives. We of course eat packed food. We of course eat tinned food. We do take medicines a number of times. So from all these different, we are drinking some different type of cold drinks and etc. So many things now. So now all these different type of chemical substances, which may be in the form of insecticides, pesticides, or various different type of chemical components entering our body in the form of some preservatives, in the form of medicine entering our body, these chemical substances should be broken down. Say suppose a person who consumes alcohol, a person who consumes so many other different type of toxic substances. So it is not that we don't consume any alcohol, that means our body is completely clean of toxins. No. Along with food also, so many different type of food items we are eating. Along with that also small amount of toxins are entering, and people who are consuming other different type of toxic substances, the toxicity in their body is even more. So now what will happen to those toxins? Even if I talk about a person who is constantly consuming alcohol and other different smoking, etc.,
It will be digested by the body.
they are not digested by the body. These toxins, by the way of, they will first of all get mixed with the blood, and these toxins, by the way of circulation, they will enter into the liver cells, and then the SER in the liver cells will break them down to some simpler forms that they can be released out by the way of the kidneys, excretion by the way of the kidneys. So if this is what is happening in our body, and yes, this is happening in our body, that's the reason, even if we are taking in food which is rich in pesticides, we are taking in food which are rich in preservatives, no? Different type of colors and chemicals, People consume so many different type of toxic substances, smoking, drinking, etc. But then also people are surviving for so many years because the liver has the capacity of breaking down the chemical substances, toxic chemical substances into simpler forms. If the liver cells did not have this property, then it would have been very difficult for us to deal with the different type of food items that we are eating. Then very soon we would have been falling sick, and very soon our body organs would have got disturbed. So this is what we are talking about is detoxification. Yes, our body already has a method of breaking down toxins to some extent. But yes, if a person is constantly year after year smoking, drinking, obviously some of the other organs will get damaged. Detoxification. And the last function that we are going to talk about would be membrane biogenesis. Now, what is the meaning of membrane biogenesis? What is membrane biogenesis? Membrane biogenesis means membrane biogenesis. Biogenesis means
And the substance formed by the
Formation.
food and the environment of the reticulum helps to build the cell or
cell membranes. So whenever we are talking about the membrane, what is the meaning of the membrane? Where are the membranes present in our cells? This is a membrane, cell membrane. Where else? Nuclear membrane. The membranes of the endoplasmic reticulum itself, the membranes of the Golgi, the membranes of the mitochondria, the membranes of so many other cell organelles itself. But if you remember, we had already talked about cell membrane. What is the composition of cell membrane? I told you the other day.
proteins and lipids.
Lipids and proteins. So now, for the entire process of membrane of a cell, lipids and proteins are required in so much quantity. Lipids and proteins are required in large amount. So for the process of formation of membranes, lipids and proteins are required. For the process of formation of proteins, lipids, for the process of formation of the membranes of any organelle or the cell itself or the nucleus, lipids and proteins. Without lipids and proteins, can a cell divide also? Not possible. For the process of division, also lipids and proteins. So that is what we are talking about, membrane biogenesis, meaning the process of formation of membrane, division of the cell, everything is dependent on the proteins and lipids released by the endoplasmic reticulum. So this is about the endoplasmic reticulum, you will remember. Next is we talk about the Golgi. Let us talk about the Golgi now. What about the Golgi complexes? When we talk about the Golgi complex now, next. When we look into the Golgi complexes, Golgi complexes or the Golgi body, Now, how do we draw? Where is the Golgi body present? The Golgi body would be present quite near the nucleus also, and also near to the endoplasmic reticulum quite a number of times. So now let us look into the concept of the Golgi here. So I talk about that the Golgi is here. The Golgi is here. And the Golgi is always in the form of some plate-like structure. The Golgi is always in the form of a plate-like structure like this. So structure, what are we going to write? It is always in the presence of, or it is always in the form of some disc or plate-like structures. It is always in the form of some discs or plate-like structure. Always in the form of some double-walled discs or plate-like structure. In the form of some network of disc or plate-like structures. Plate-like structures. And these plate-like structures are always stacked. These plate-like structures are always stacked one after the other. They are always stacked one after the other. Okay. Now, this Golgi, do they have any types? No, not exactly. They don't have any types as such. Now that we have been talking about the endoplasmic reticulum, they don't have any types. But
they have it.
Huh?
They have it.
No, types not. But this Golgi, these tubules that we are talking about, the stacks that we are talking about, sometimes they may take different shapes. But as such, when we talk about the types, no, types actually they don't have. So now, whenever we are talking about this, they may sometimes be flattened. These may be sometimes flattened sacs. Sometimes flattened sacs or tubular sacs. They may be sometimes flattened. They may be sometimes tubular sac-like structures, etc. So that is what we are talking about. But they will not be that. That is not actually considered as the true types. So they are flattened. They are tubular. Now, next is that when we are talking about the Golgi, the sac-like structures, what are they termed as? The sac-like structures are known as the sac-like structures are known as most commonly cisterna, singular cisterna, most commonly cisterna or cisterna, and they always give rise to what? They will always give rise to vacuoles and vesicles. They will always give rise to vacuoles and vesicles. So that is their major feature. Cisterna, you are always asked sometimes on mark. Cisterna, what are cisterna? These are some tubular structures. These are some stack-like structures, sac-like structures. This is what is called cisterna. Now, what is their function? What are these Golgi doing? This Golgi, now that we talk about the Golgi bodies or the Golgi complexes, are majorly meant for doing what? The Golgi bodies or the Golgi complexes are majorly, they are engaged in the secretion of variety of enzymes, almost 40 different types of enzymes, proteins, variety of different types of enzymes, proteins, etc. So they are engaged in the process of formation of variety of different types of enzymes, proteins, hormones, etc. Now, here some other function that we are going to talk about. They also help in packaging of cell secretions. They also help in packaging of cell secretions. Now, they also help in packaging of cell secretions, meaning, now say suppose I talk about that this is a endoplasmic reticulum, okay? This endoplasmic, what type of endoplasmic reticulum is this? Rough. So now this vesicle is containing what? Supposed to contain what? Proteins, no? So these proteins will come and move towards the Golgi. So what is happening? It comes and fuses. This vacuole is coming and fusing. This vacuole comes and fuses with the Golgi. Check here. Now, as it comes and fuses with the Golgi, what happens? The contents of the Golgi is entering into the Golgi. The contents of the endoplasmic reticulum is releasing into the Golgi. So what I told you, this is a small vesicle containing secretions. This is a small vesicle containing proteins from the endoplasmic reticulum and moving towards Golgi. Why should it move towards Golgi? It is always a process where the rough or the smooth endoplasmic reticulum, whatever molecules they produce, directly they cannot undergo the process of function. Directly they are not ready for any function. They have to be ultimately processed and packaged first. So where will the processing and the packaging be done? In the Golgi. So what are we doing? Just for example, you talk about any type of a factory which is preparing the fruit juice. Talk about those farms where the cows and the cattle, they have been milked. So what will happen? First they will collect cans and cans of milk, and then this milk will go to some other factory, for packaging, and then the packets of milk will come to the shops and then to our houses. So there's a particular route by which the milk has reached our own homes. So exactly like this, for example, this is a farm which is involved only in the process of secretion. Now the protein is produced, packed in a small vesicle. But are these proteins actually ready to show any function? Not yet. So they have to go towards the Golgi, empty their contents into the Golgi. Then what will the Golgi do? The Golgi will again pack it up after processing. The Golgi will again pack it up in another particular vesicle and then ultimately release it off. Now this vesicle which contains the enzymes or the proteins are ready to show any function.
So does Golgi add any more proteins and enzymes?
Golgi basically produces its own enzymes, proteins, hormones and packages that and releases also, and it also will package secretion.
The secretions that are produced by the
endoplasmic reticulum. It is just modified. It will just process, meaning little bit of some the composition will change. but it produces its own also, and it helps in the packaging of this also, cell secretions. So what did it do here? Now it is releasing so many different types of such vehicles. So many different types of such vehicles are released. Vacuoles are released. So now that I talk about that these different type of vacuoles that the Golgi is releasing are in so many types. See there, we are already talking about that the Golgi can produce almost about 40 different types of enzymes. Golgi can produce 40 different types of enzymes it can produce, right? So Golgi can produce 40 different types of enzymes. That means Golgi can produce this type of vacuoles on its own and package this. Now the question is that these different type of vacuoles that we are talking about, what would be the job of this type of enzymes or the substances? Packaging of cell secretions. Now here we talk about it also helps in the formation of the lysosomes. It also helps in the formation of lysosomes. Now why are we talking about lysosomes? That's a separate organelle altogether. We are talking about lysosomes only because these vacuoles just now that I told, which contains proteins or enzymes, out of the variety that it produces, there may be one type. Out of the variety that it produces, there may be one type which possesses a specialized type of an enzyme. And what are those specialized type of enzymes doing? They can bring about digestion. Digestion of any organelle, digestion of any digestion here means dissolving. Digestion of any organelle, digestion of any membrane, etc., etc. So this is a specialized type of a vacuole that the Golgi has produced. Again, the Golgi produces such type of vacuoles in huge numbers, many categories. So this is one type, a specialized type, and I would say that this is what is known as the lysosome, because the lysosome is a specialized type of a vacuole formed from the Golgi, which contains hydrolytic enzyme. What's the meaning of hydrolytic enzymes? Hydrolytic means which can bring about the process of breakdown and digestion. So this is about the process of, this is about the concept of Golgi. Now, with this continuation, now if I talk about lysosomes, I'm talking about another organelle now. I'm talking about lysosomes now. What about the basic structure of the lysosomes? What would be the basic structure of a lysosome? How about its membranes and all? It is single membrane. It has got a single membrane, single wall it is. It has got a single wall, single membrane. What is? It contains hydrolytic enzymes. It contains hydrolytic enzymes, hydrolytic, digestive, hydrolytic enzymes. Okay, so that is one. And then the small lysosomes or primary lysosomes, small and primary lysosomes fuse together. Small such lysosomes, which are known as primary lysosomes, will fuse together to form large lysosomes. And what are those large lysosomes known as? These large lysosomes are known as secondary lysosomes. What are they called? These are called secondary lysosomes. Secondary lysosomes. So here I am talking about two types of lysosomes. Primary, which are the small ones, just formed. And then two primary will again club together. See, suppose I may talk about two primary clubbing together and resulting in the formation of a big lysosome here. So this is a secondary lysosome that we understand. Okay. Now, function. Then what are these lysosomes actually doing? What are these lysosomes actually doing? If I have to talk about the function of the lysosomes, function of the lysosomes would be, number one, can help in the digestion or breakdown. Digestion or breakdown of the organelles of the old and worn out cells. Hence it is known as the suicidal bands. Hence it will be termed as suicidal bands. Why is lysosome known as the suicidal band? So this is what you'll have to remember. Hence it is known as the suicidal bands. Okay. So why is the lysosome known as the suicidal bags? What is the job? What is its role? So the lysosome basically contains a specialized type of enzyme. What is that enzyme? What is the category of enzyme known as digestive or hydrolytic? So now, when a cell is already old, finished, function is finished. Now it cannot divide. Now it cannot show any function. What will happen? What should happen to that cell? That cell should break down. That cell should break down, and that cell should get released out of our body along with our waste substances. But unless and until it is broken down and converted into some simpler forms, how can the process of release happen? So that job is done by the lysosome. Say suppose I talk about the mitochondria, I talk about the chromosomes, I talk about the nucleus. The lysosome will just go and get fused, and the enzyme will just act over and then dissolve everything. Okay, so that is the role. If that is what is the lysosome doing, then the lysosome is always known as a suicidal bag. The lysosome is known as a suicidal bag. But will the lysosome ever bring about the digestion of the cell organelle of a functional cell? Will it ever bring about the digestion of the organelles in a cell which is going to divide and well functional, synthesizing enzymes? No, not at all. Only when the cell has completed its functions and about to die out and about to become non-functional, only at that time. This is a concept quite often you are asked why are lysosomes known as suicidal bags. So now we are already
That lysosomes, what is the
primary lysosomes fuse together to form large lysosomes called secondary lysosomes. Large lysosomes called secondary lysosomes. Okay. So we had already talked about the major cell organelles. Which are the cell organelles we had already covered up? The nucleus. Nucleus we finished? Nucleus we did not finish. Okay, nucleus we had finished. Mitochondria we had finished.
Yes.
Then we had finished with cell membrane, cell wall. No? Mitochondria done?
Yeah, yes.
Endoplasmic reticulum we have done today, and similar type of organelles. 3 similar type of organelles we finished today. Endoplasmic reticulum, Golgi, and lysosomes. Okay, now we talk about the nucleus. About the nucleus, now the concepts are absolutely different when we talk about the nucleus. Now, in this case, again, let us go back to the structure of a nucleus only. Let us go back to the structure of only the nucleus. Now, to talk about the structure of the nucleus only, let us see how would the structures be. If you remember, how did we draw the structure of the nucleus? The nucleus always has a double-walled membrane like this. The nuclear membrane, which is known as the nuclear membrane. A double-walled membrane, it is a double-walled membrane. Now, this nuclear membrane inside, what is there? A whole lot of coiled thread-like structures known as the chromatin fibers, and then a spherical structure known as the nucleolus, a dark stained spherical structure known as the nucleolus, and some cytoplasm inside. So what are the parts here? This is the nuclear membrane, nuclear envelope, or the nuclear membrane. Next is, remember, the nuclear membrane has got pores. The nuclear membrane is not continuous, it has got pores, nuclear pore it is known as. Then what about this cytoplasmic part? We will say cytoplasm is known as the nucleoplasm. The cytoplasm is known as the nucleoplasm. What about these thread-like structures? These thread-like structures are known as the chromatin fibers when they are very long and especially in a very small cell. But when they are mature, when the cell is mature and the chromatin fibers are extensively coiled, then we will say that these are chromosomes. These are no more chromatin, but chromosomes. And what about this dark stained structure? This is known as the nucleolus. This is known as the nucleolus. So what are the 4 parts which make a nucleus? The 4 parts which make a nucleus, nuclear membrane. So what should the nuclear membrane be made up of?
Proteins and lipids.
Correct. Proteins and lipids. Then we have got the nucleolus, then we have got the chromatin and the cytoplasm. The cytoplasm and the nucleoplasm more or less the same, except for certain specialized proteins which are present. Yeah. Structure, if I have to understand some properties already I told you about the nuclear membrane and the nucleoplasm and all these I'm not repeating. So for the structure, majorly we talk about the chromatin fibers. When we are talking about the nucleus, what does the nucleus contain? They would contain the thin thread-like structures known as the chromatin fibers. Now, these chromatin fibers, if I see, carry what? These chromatin fibers, they carry hereditary information. They carry hereditary information. So whenever we are talking about the chromatin fibers, these are nothing but fine, long, thread-like structures. Fine, long, thread-like structures. These are chromatin fibers carrying fine, long, thread-like structures. So these chromatin fibers, which carry fine, long, thread-like structures, always are known to carry the hereditary information. But if I talk about that, what can be the relation between this chromatin and the hereditary information? The relation is, I am talking about a very long thread-like structure. I am talking about a very long thread-like structure. But each of these thread-like structure, when I see that they are extensively condensed, when these thread-like structure, when I see that they are extensively condensed, now this is what I am to give a different name, and this is what I say, that this extensively condensed chromatin fiber is what are known as chromosomes. Then what is the difference between the chromatin fiber and the chromosomes? Chromatin fibers are very long, very thin. Chromosomes are extensively condensed. I will give you the answer, don't worry. Chromosomes are extensively condensed. Now, I want to find out a small portion of these and see in detail how does it look like. How does it look like? Now, I get to see that if I magnify and try to see the internal structure of a small portion of the chromosome or a small portion of the chromatin, I get to see that the structure is almost like this. I get to see that the structure is almost like this, where it is a ribbon-like structure, here. And this ribbon-like structure has got some basic structural aspects, and it almost looks like a ladder. Where there are a whole lot of units of which it is made up of, various different type of components of which it is made up of. A little bit of it you will learn in the 10th. So as of now, we are only to understand that what is this chromatin fiber or the chromosome made up of? It is made up of a ribbon-like structure, which is made up of 2 threads. Can you see? It is made up of 2 threads, and that's the reason it is known as the double-stranded DNA. Double strand means 2 threads. Double-stranded DNA molecule. And this double-stranded DNA, when I say, what is the full form of the DNA? You have to remember, the full form of the DNA is deoxyribonucleic acid. It is a complex acidic molecule which is made up of so many different type of components. I go back to this concept. Why did I write that the chromatin or the chromosome contain hereditary information?
As DNA has the gene cover parents, both the parents. Like the child also like looks, talks, or might be anything else.
Exactly. When we are talking about hereditary information, what is the meaning of hereditary information? We have inherited characteristics from our parents. Our parents have got characteristics from their parents. No, that is how generation goes on. So now, what is how do we understand hereditary information? I would say that here, when I talk about this particular DNA, a small section of the DNA, maybe this is responsible now. This small section of the DNA may be responsible for our hair color, and another section of the DNA I may talk about. This section of the DNA is actually responsible for our height. Another section of the DNA may be responsible for our skin color, etc. Nucleic acid, NuCleic. NuCleic. So now, each of these segments of the DNA, the entire DNA can be divided into small segments, and every segment
has different function,
controlling one particular character that we show. One segment for our height, one segment for our skin color, one segment for our hair texture, one segment for the shape of the nail, one segment for the shape of the eyes. It's like that. So what are these segments called? These segments are called the genes. What are these genes? These segments are called the genes. Then what is the definition of the word gene? The functional segment of the DNA. The functional segment of DNA the functional segment of DNA that regulates or controls. We can write controls, a common word. So, functional segment of the DNA that controls a specific character. That controls a specific character. What is the meaning of the word character? Are features. Is it only character that we understand when we talk about the basic concept of English? When we talk
Those are characteristics.
characters or characteristics, it's the same thing. You can write characters, you can talk about characteristics. So when I talk about characteristics also, say suppose when I understand characteristics, what is the meaning? Simple English meaning of characteristics is what? When I talk about the characters or characteristics, basically English meaning would mean how is our behavior, whether we are rude or polite or short-tempered or cool, exactly this way. But in the concept of biology, characters actually does not mean this. Characteristics means each and every bodily feature. The shape of the eyes, nose, ear, the color of the eyes and the skin and the hair. These are all characteristics. We talk about even the enzyme and the hormone present in our body. That is also the character or the characteristics. So here we are not only talking about a concept of our own behavioral or personal characteristics, but everything related to our bodily features, enzymes, hormone, pigments, etc. So hereditary characteristics. You are asked, name the structure which is responsible for the transfer or the inheritance of characters. Then what are you supposed to write?
DNA.
Whenever you're talking about the structures which are involved, structures which act as carriers of heredity, structures which act as carriers of heredity, chromosomes, and chemical components
DNA.
which help in expression of a character, DNA. Chemical components, because the DNA is nothing but a big chemical molecule actually, it's a chemical substance. It's a big complex chemical molecule, the DNA that we talk about. It's a big complex chemical molecule. Now, whenever we are talking about the the nucleus is also known as what? It is also known as the director of a cell. Why? Because now so many organelles we have already studied, no? So many organelles we have studied. What are the organelles we have come across? Mitochondria, endoplasmic reticulum, lysosomes, plant we have to start. Now, all these organelles so far, whatever you have studied, are under the control of the nucleus. The nucleus will give command what type of functions, what type of proteins, what type of enzymes to produce. But among this, we have studied one organelle which can still control some of its functions on its own. That is the mitochondria. Why? But
because it has a DNA.
It has its own DNA and it can produce its own proteins. It has its own DNA, so it is not fully dependent on the nucleus. It can keep on synthesizing its own proteins and it can keep on dividing its own, show various functions on its own. So this is all about the animal cell basically that we have to study. Animal cells, what are the prominent cell organelles? Now we have to also talk about the plant cells. But before we go into that, take a check if you are thorough with this part first. You got pencil. Take a check if you are thorough with this part first, then only we move on to the next section, that is the plant cells. Try these three questions. Page number 88. Check this. Page number 88, question number 256. Page number 88, question number 256. Did you get that? Did you get that? 256. Page number 88. 256. 5, 6. Let us try solving some of these questions. Write down the question in the notebook. Huh?
Yes, but already.
No, some you have to write. You have to do enough writing work. 2, 5, 6, I said. Board question, yes. These are all previous year questions from different exams. Write down the name of the chapter, write down the questions. Right? No, here only. Write down the question. What is the first question? Just check.
What makes lysosomes such effective digestive bags?
Okay. So just listen to the questions. We'll just discuss it out and you will do it as homework. The first question is what will you be able to answer? What makes lysosomes
Such effective digestive
bags? What to write there? They are considered as effective digestive bags because
of the
presence of hydrolytic enzymes, because they contain hydrolytic enzymes and which can break down old and worn out cells, which can break down old and worn out damaged cells also. Old and damaged cells, old and worn out cells. Okay. Come to the next question. You will try this as homework. Proper answer you will write in 2 or 3 sentences. Number 2, देखो फटाफट go through if you are able to answer.
What is fundamental unit of life or discovery? How can it be observed?
So if this you will be able to answer. What is fundamental unit of life? Cell. Who discovered? You know.
But
What is the third part?
how can they be observed? Electron microscope.
How can they be observed? No, they can be observed under the compound microscope also. Cells can be observed under the compound microscope. So how can they be observed? They can be observed under the microscope. They can be observed under the microscope, compound microscope, detailed structure they can be observed. What is the next question?
Can a single cell live independently on its own? Explain by giving an example.
Yes. Yeah, how? Example give me.
Mitochondria.
Single cell.
Single cell.
Mitochondria is not a cell.
Amoeba.
A single unicellular organism is studied. So how can a single cell live on its own independently? Yes, it can. It can show all its functions and it can divide also to give rise to new generation cells. For example, unicellular organisms like amoeba. Try framing these answers. If not, if you have a difficulty, then again the next class we will do this. Next class again we will go through some more questions, complete with the plant cell part, and the major concept of this chapter is still left, the process of osmosis diffusion that we have to study.
क्या करके रखती है? ये भाई देख लिया मुझे कुछ नहीं समझा।